Okay, back to our graph, making sure that we put in our standard deviations properly. You're going to select the column that you are going to add the first standard deviations to. You will go ahead and go into the options, and you can do that by going to add chart element, error bars, more error bar options. This is your baseline data set, or is it your undifferentiated data set? So you're going to go into custom. Make sure you look at what it is that you are selecting. So since this is my undifferentiated set, you are going to select for the row which contains the undifferentiated percentages. You are going to select the standard deviations both for the positive as well as for the negative side, and then click on OK, and it will automatically put those values for you. You can repeat that process for the rest of your error bars. So here now I have selected the next one, which is stage 1. Again, go to customs. Now you're going to go ahead and select the stage 1 numbers for all my individual treatment groups, selected for both plus as well as minus, and it is going to select those for you and add them in. Repeat that process until you get to all of your bars. So since we only have 2 more, I'm going to show you. Okay, and then the last one that you've got, custom, specify value, go ahead and select the last row, and you have your graph essentially finished with all the appropriate material in there. The only thing you need over here is the axis title. You want to make sure it is labeled properly. So I'm going to add the title in here as percent cells in each stage. You can have a slightly different title, whatever you want, that refers to the graph that you are showing in an appropriate manner. When you're happy with the graph that you've got, you can copy it into your document and then add copy. And for example, if I was here, I'm just showing you an example with a new slide. You can then go ahead and paste it in here, either as a picture or as a document, and add a caption and a title and caption underneath it, starting with figure 1A, whatever it is that you want to talk about. Make sure the result text, the narrative, is detailed enough that I do not need to necessarily see the table or graph to understand what it is that you found. Also, the actual caption and title of your figure should be detailed enough that I don't have to go to the result narrative text in order to understand the graph or table that you're presenting either. So that's your figure 1, or one of the figures that you have to draw for the cell differentiation paper. Now comes the part where you're going to do the statistical analysis using this particular data set. You are not comparing all the various stages with each other. You're only going to be looking at the percentage of cells that have differentiated. In many papers, they are defined as stage 2 and 3 combined. You can also look at just stage 3 if you are in a time crunch. However, since many papers do talk about stage 2 plus 3 combined, I do think that it would be better choice to use those numbers themselves. To do that, you're going to essentially do a process similar to what we were just doing for our work right now, except this time you're going to first add the 2 numbers that you've gotten up here together. So I'm going to go ahead and make a separate area for that down here, where I will say that these are my percent cells that are differentiated, and you want to make sure that you let me know what you are using. If you're doing stage 2 plus 3, then you want to tell me that it was stage 2 plus 3. If you're doing stage 3 alone, then you want to let me know that it was stage 3 alone. And then this time you're just going to have your different treatments, in my case the baseline, you know, high serum control, low serum control, and whatnot. So you'll just have your 5 treatments, and you don't have to worry about having the various stages now listed, because you're just going to have that one number. So LS, DMSO, and RA. And then the first thing you need to do is obviously to first get the numbers the way they are over here for each one of the groups individually. You cannot have the data with just the averages. You need to have it for all your groups. So G1, G2, G3, I only have 3 groups. I can just do it with 3 groups. I'm going to start by adding my stage 2 plus 3. So I will say stage 2 plus 3. Oh, I need to go all the way up here actually to the baseline. So I'm going to say stage 2 plus 3. And then I'm going to divide by total number of cells, just like I did before, and then multiply by 100. And I should be able to get the number that I need. And there you have it. You're able to get the number. And then you can pull over to get the number for the remaining groups, no matter how many groups you have. It could be 3, it could be 100, but all the groups you have can be used together in this manner. Okay, so now I've gone ahead and calculated that information for the group 1. I can drag it over and I have that information for everyone. Now I am ready to compile the data for my actual analysis. I can do the average again, so say average and copy the numbers over just like I've done before, no different. So it shouldn't be a surprise or a difficult thing to do at this time. You're doing the exact same thing. Make sure you get your standard deviations the same way, because obviously it's no good if you don't have that information. And again, select what you need to do. Go ahead and get your values. You can create the graph just like the one that you've done before with this information, where you show the data, the treatment groups and the averages, and then input the standard deviation. However, for the purposes of the t-test or the ANOVA, which is what you will use for statistical purposes, you need to do the analysis now. So I'm going to go ahead and show you how to do the t-test first. To run the student t-test, using the simple Excel method, what you need to do is you need to know what the variance is. You can calculate it using the statistical packages that it provides. I'm going to show you first just to run the statistical portion of it, which is the actual t-test itself. And then there are links posted to your Blackboard which show you how to calculate the deviations and all that good stuff. So I'm going to go ahead and copy my copy these treatments, so I can go ahead and start